Hi and welcome. This is a pre-conference workshop video for the 2021 West African Society for Pharmacologists Conference, which is hosted in the Department of Pharmacology and Therapeutics, ABU Zaria, and was prepared courtesy of the Molecular Modeling and Cheminformatics Drug Design and Synthesis Research Group from the Department of Pharmaceutical and Medicinal Chemistry, Ahmadu Bello University, Zaria, here in Nigeria. The workshop would, During the course of the workshop, we'll be taking a look at the use of molecular docking in drug discovery, where we'll be exploring the concept of dock validation, virtual screening, and postdoc analysis. The necessary files and the guide materials that will be used are available for download at the link provided in the details section below. And the guide material contains link. This is the guide material which I've opened here. It contains links to download our target, important files I'll be using, and the various software that I will be using. These softwares include Chimera, Discovery Studio, Autodock Tools, and Cygwin. If you run into any issues, you can contact us with the link provided, or you chat with us on WhatsApp. Um, First off, we should download the important files in this doc folder, and we can go to our web browser and click on that link provided. It opens up this page for you, and you can click on download right here. By clicking on download, the file downloads in a zip format, and we can open the file and we keep them here. We go back to our material, and then the first thing we have to do is to get our targets. To get the targets, the crystal structure that will be used for this is GABA AAR, and we'll be accessing it from the RCSB protein data bank. So all I have to do is copy the link here and navigate to my web browser, and I open up the home page. On the home page, I can type in the PDB ID or the name of the particular target I'm interested in. I have the PDB ID and it's 4COF. You can search for it. It's telling us that we have the crystal structure of human gamma amino butyric acid receptor, and the information that are necessary: the organism, the expression system, the mutations. It contains no mutations, which is good to go. We can, if you're actually interested in the mutant type of the target, you'll have to search for it and access it also. This was the date the enzyme was deposited on the database, and we can see that if you scroll on down, we get to see the literature, and you can read the abstract, and you can download the journal also from Sci-Hub using the DOI. Scrolling on down, we see more information about our targets, about the macromolecule, and you can see that it has about 5 chains. The Uniprot ID, which is also a database, and then we scroll on down, we see the small molecules. These are the various small molecules that are present, of which benzamidine is of interest to us, as it will be our native ligand during the course of this study. The next thing we want to do is create our doc folder, and it's best to create it on the desktop. So, and we can name it anything. So I'll go ahead and create a folder on my desktop. So I'll go ahead and name it, I can go ahead and name it docking folder. Here is in caps lock, so I know that's the folder I want to save it as. Doing this, if you remember, we have our files that we downloaded from the doc folder online here, and I want us to extract vina, extract the conf file and the vs.bash file. You hold on control and select them. I will extract to the folder that we just created, which is on desktop, and you can see it's docking folder, the one in caps lock here for me. It could be any name, whatever name you used to save yours. I'll press OK. I can go back and check the folder. This is it here, and you can see that the files are there already. This is the folder where I'll be saving everything that is related to these docking studies here. So moving on, the first thing we want to do is to prepare our receptor and our ligand for docking using Chimera. Contains the newly downloaded target from PDB. To download our targets, we scroll up and we go to download files, PDB format. We see that the file will download in our downloads folder, and then we can navigate to the downloads folder, copy the file, go back to the desktop folder and open the docking folder and paste the target in it. By so doing, we are ready to carry out the first step, that is the receptor and ligand preparation using UCSF Chimera. And these are the steps that we'll follow. We'll open UCSF Chimera, and then we open the newly downloaded target. Click on open, we go to desktop. The folder is docking folder, and the file is 40f.pdb. It opens up the target. As you can see, it's quite a big target. From literature, we'll be needing only chain A, and the only ligand that we'll be needing also will be benzimidine. So it gives us a clue to reduce the bulk of this target, reducing it only to chain A. Then downloaded targets can also be remodeled on Swiss model for targets that have incomplete residue. You can check that out and try remodeling any targets that you notice that doesn't have a complete residue. Moving on to select, we want to get rid of every other chain except chain A. We go to chain, we click on A, and then we want every other thing here gone apart from what we just selected. So we go ahead and we'll choose invert selected models. It selects every other thing that we didn't select initially, and then go to actions, atoms or bonds, delete. This deletes them. You can use your scroll button, press it down to move your target around, and you can use the scroll button up and down to zoom in and zoom out respectively. The next thing we want to do now is prepare our enzyme for the docking studies, and we'll go ahead and delete all non-standard residues. Come to select residue, we select all non-standard and select every other thing that is not part of the standard residues that make up this enzyme. I will click on actions, atoms or bonds, delete. This gets rid of them and we're left with only our enzyme here, or our target. The next thing we have to do is go to tools, surface or binding analysis, and we have our dock prep. Here we'll be preparing the enzyme for docking by adding hydrogen, adding charges and the likes. Since we don't want our file to be in mol2 file, because we have an enzyme, a big file here, and mol2 will not support such. Mol2 supports small ligands with their charges. We'll uncheck write mol2 file and we'll click on OK. For the add hydrogens window, we leave it as it is default, and we'll click on OK. Then for the assign charges, we make sure we assign Castilla charges, and we'll click on OK. And it has finished preparing it. As you can see, it wrote doc prep finished. Then we'll go ahead to file, and then we save the PDB file. We navigate to that folder that we created for the docking studies. As you can see, mine is the one in capital letter, docking folder, and we can decide to name it rec. That's receptor, REC, rec, and we'll click on save. It will save it as rec.pdb now. Then is to prepare our ligand also for docking using Chimera. We close the session, go to file and close session. Then as you can see, it's readily accessible here. The file we opened initially, that's for our targets from PDB. We click on it, force COF, and we do the same procedure where we delete the chains that we don't need, invert selected models, actions, atoms or bonds, and we delete. Then now we also want to leave only benzimidine. So do the same procedure, go to residue, click on ben, we click on select, invert selected models, actions, atoms or bonds, and we delete. Now we are left with only benzimidine. To prepare it for docking, we go through the same process, go to tools, surface or binding analysis, and we can click on dock prep. Now we want to check the right mol2 file because it's a small molecule, and in case it has charges, we'll be able to save the charges along with it. So we'll click on OK. We leave the add hydrogens segment as default. Then we ensure that we add Castilla charges also as well. We take note of the net charge, and in this case is 0, so we are good to go, and we'll click on OK. Then it prepares it, and then it pops up the window where to save. So we make sure we navigate to that docking folder that we created for our study, and then the file name, we can name it as lig, and then we click on save. With that done, the next step is to get the residue numbers of the active sites using Chimera as well. We click on open, we click on file, and we click on close session. We open the rec that we prepared, and we click again file, open. Now we open the ligand that we've also prepared, lig.mol2. Then the next thing we want to do is we want to check the enzymes that the active residues, the residues that make up the active site around benzimidine. So we'll click on select, we click on select, we click on residue, and then we choose benzimidine. Then we go to select, we click on zone less than 5.0, and we click on OK. It highlights the various residues that are around this particular ligand. By so doing, we can go on to tools to check the residues, and under tools we go to sequence, and we click on sequence. It pops up the sequence, and we should take note of the sequence of the amino acids, the ones that are highlighted in deep green, as these are the residues that are at the active site. You put your cursor over it, and you can get the name of the particular amino acids. This is LU99, this is GLU155, this is TYR205. You can go ahead and write them out in notepad or on a piece of paper, and then you can close this when you are through. You can then save this ligand and this receptor as a complex. Go ahead to file, save PDB, then we check save multiple models in single files. So say we make sure it's in a single file, and we can rename it to com. Make sure it's saving in the docking folder. Click on save. Now we'll move on to dock validation. For the dock validation, these are the steps that are involved. We can close the session here. Then we open our rec again. And then we also open the lig.mo2 file. Then we go ahead to favorites, model panel. Then we click on the rec. We scroll down here to rename. And then we can name it as rec.pdb. We check the rename models box and we click OK. Then we click on the second model. Then we scroll down as well to rename it. I will click on rename. Now this time around, name it as league.mo2 since that's our ligand and it's the second model. We'll click on OK and then we'll click on close. Then we'll go ahead to tools. Then we'll go ahead to surface/binding analysis and we'll click on Autodock Vina. For Autodock Vina, we have to choose where the file where our output will be saved. So click on browse. Make sure that it's in that same folder and you can just name the file, call it data1, and you set output location. For the receptor, you click on it and you click receptor. For the ligand, you click on it and you click your lig.mo2. Make sure you check the resize search volume using a particular button. You can decide to change the button. Let's say change to button 1. You can come out there, zoom in and try to draw your box over the ligand. In the windows that pops up here, you see it has created the centers values and the size of this box that we just drew. This is the active site. You can then hover your mouse on this and try to reduce the size till it's compact enough and adjust the box where the box is. For the purpose of this workshop, the active site has been defined where we have our center X, center Y, Z, the size. So we can go ahead and copy this in and paste them there. So open Chimera here for this. Then for center Y, then for center Z, put it there. Then the sizes are 10, 10, and 18. So put 10 in here, 10, 18. We click, we are okay with that. And if you check our box here, we can see it has put a nice, good looking box around the target in this active, around the ligand in the active site. We click on receptor options and leave as default. For ligand options, we leave as default. For advanced options, you also leave as default. Then for the executable location, we want to check the local option and then we'll click on browse. Then we navigate to the docking folder. If we open our docking folder, we can see that we have the various files there. We'll open Vina and then we click on vina.exe and then we'll click open. We'll click on apply and we'll wait for it to run the docking. As you can see, it's running our dock right now. As you can see, it has finished running and we can see our new molecule is there. Soon as the docking study is over, we can click OK, and then we can start checking the various poses. See the various poses that the new ligand that we just docked in is giving us. We check for the superimposed structure and ensure that this compound that we just brought in is well superimposed, at least you can see, with our particular native ligand. The first pose usually being the best one with 0.0 RMSD values. The superimposed docking studies telling us that we have gotten our active site well defined and we are good to go to carry out virtual screening and the likes. But before we go on to carry out our virtual screening, we'll have to do some postdoc analysis using UCSF Chimera. These are the steps that are involved. Opening UCSF Chimera. So we can close this session and then we can go ahead to open our rec.pdb. After we opened our rec.pdb, we go ahead to tools, surface or binding analysis. Now we want to view the native ligand that we just redocked into the active sites for our dock validation. We want to see the kind of interactions that occur there. So go ahead to view dock. It opens up the folder, and don't forget we saved it as the folder as that point pdb qt, and we click open. We see that it brings out our prepared and our docked compound here and the post that is add, giving us the docking score here as minus 5.3 for the best. Next is minus 5.2. You can go through like that. Then we want to visualize the hydrogen bonds. We go to select, then we choose our residue, to choose the ligand, that's BAN. Then we go back to the view dock window and we click on hydrogen bonds. Click on the first option there, and then we click on check intermodels. We check our intermodel, then we check the label hydrogen bond with distance. This helps us to check the distance between the respective residues and the ligand. Then we should also check the if endpoints atom is hidden, it should show us the endpoint residue. We click apply, select, and then we clear selections. We click apply. As you can see, it adds more information here, and then it shows the hydrogen bond that we have and the distance that the hydrogen bond is having with the receptor, between the receptor and the ligand. We can go to select and we can clear all selections. We can then save this as an image. Saving it as an image in the same folder, we can name it as image and then we can click on save. It saves that as an image for us. Then we can always change the background color. That is, if you're interested in changing the background color, go to favorites, preferences. Under preferences, we click on general, we click on background, and can leave it as solid, gradient, whichever one we want. Then background color, we'll move the sliders, we'll click on them and move them to our desired position at least. Moving them to the extreme right gives you a white background, and you can save. You can close this, and then you can go ahead and save this as an image. If you're interested in saving with a black background or white background, so image one, and then we click on save. With this done, we can close this box. Then we can also play a movie. We can play a movie of the possible poses. So we'll click on movie, I'll click on play, and you see it starts showing the various possible poses that it has and the bonds, the hydrogen bonds that it has for each pose. Soon as we are done with this, we can stop the movie, and then we can save the complex that we want relative to the pose that we're interested in saving. We'll be going for the first pose. So to save that, we click save PDB. Then we make sure we check that save multiple models in single file is there, and then we'll click on rec.pdb and we'll press down Control and click on number 1.1. That is the first post that it showed, the one that had 0 RMSD values, and we can rename it as, for example, rec binded, and we click on save. We can also go ahead and view the hydrophobic surface of the complex. So we close this session, we go to our recbinded.pdb that we just saved. Then we go to presets, then we click interactive 3, that's hydrophobicity surface. By so doing, we wait for it to convert it to the hydrophobic surface, and here you go. You can see giving us the hydrophobic look of the enzyme and our ligand benzimidine there. You can always save as an image or you save it as a PDB file, just like we just did. Moving on, we'll go on to postdoc analysis using Discovery Studio. For postdoc analysis using Discovery Studio, we open the Discovery Studio folder. We open the Discovery Studio software. We go to File, Open. Then we navigate to that doc folder on our desktop, docking folder, and we choose the complex of choice. We could go for com.pdb, or let's go for the one we just reduct, that's our recbinded.pdb, and we'll click on open. Then we go to receptor ligand interactions. Here we can step through ligands, and stepping through ligands happens to be the only ligand there, so you can see it has shown us that it is benzimidine, and then we can then show the 2D interactions, clicking show 2D diagram, and it shows us the various interactions with the legend below. We can see it is having van der Waals interaction with THR202, GLY158, TYR97, SER156, TYR205, and the conventional hydrogen bonds with CYR157, GLU155, and it's also having a pi-pi stacked interaction with PHE 200. Then we go to the concept of virtual screening of multiple ligands. And here it is explained that we can follow the same steps to dock our different ligands in the active site, but this can actually be um, time consuming if we have a large library of compounds. So to make it easier, we make use of Autodock via a terminal such as Cygwin to dock multiple ligands consecutively. But before we can do that, we have to create PDBQT files of our various ligands. So to get the ligands that we'll be making use of in this to the zip file that we downloaded, I have I've placed some ligands there for us to make use of so we can extract them also. That's ligand 1, ligand 2, and ligand 3. You can also use ligands of your own design and choice. Then we extract them to that docking folder that's on the desktop, and then this docking folder here, and I'll click on OK. If I check the folder, we can see that those leagues are there now, league 1, league 2, league 3. They are in Mo2 format, but we have to convert them to PDBQT format using Autodock Tools. So we convert our receptor to PDBQT, and we convert our ligands, the various ligands that we have, to PDBQT as well. Open Autodock here, we come to File. To convert our receptor to PDBQT, we have the read molecule, file read molecule, then we navigate to that folder that contains all our files, docking folder, then we choose rec.pdb. It opens it up on Autodock. Then we choose grid, we choose macromolecule, then we choose the macromolecule as rec, and then we select the molecule. This converts it to PDBQT, found 2178 non-polar hydrogens, and it has merged NPH. So we go on to save this as rec.pdbqt, but we'll make sure it is also in that docking folder, that is rec.pdbqt, and then we'll just go ahead and click save. As soon as it is saved, we can right click on it and delete it. Then now we want to prepare our various ligands, so go to File, Read Molecule, then we can select them using Control and clicking on the molecules we want to prepare as PDBQTs, the native ligand, the lig 1, lig 2, and lig 3. We click on Open, it pops them up. Then we can choose ligand inputs. Then we choose for the native ligand, we press this, we select the molecule to dock. It pops up this box, we click on OK. Then we go back to ligand, output, save as PDBQT. Then we'll make sure it's also in the docking folder that we are saving it as, that's lig.pdbqt. Then we have lig 2. Lig 1, we do the same step for lig 1. We go to ligand, inputs, choose lig 1, select OK, ligand, output, save as PDBQT. Make sure it's in the docking folder, then we save. Then lig 2, ligand, input, choose lig 2, select OK, ligand, output, save as PDBQT, save. Then for lig 3 also, ligand, input, choose lig 3, select Okay. Ligand output, save as PDBQT. Make sure you save in the folder. So we can go back to our folder and check for the PDBQT files. We can see we have the lig1 PDBQT, lig2 PDBQT, lig3 PDBQT, and our receptor is also in PDBQT format. Then we have to dock our ligands into the active site of the receptor using terminal, and this terminal we're making use of is our Cygwin terminal. The conf.txt file that we downloaded earlier contains the right configuration, but if you are to make use of it for another docking studies, you have to make sure that you open it up, then you type in the right values for the center XYZ and the size XYZ. Since we have that sorted here, the next thing is to open our terminal, and then we'll print the working directory of the dock folder. We navigate to the dock folder, and then we'll run our docking studies. Opening Cygwin, the local disk, using the command cd space c then colon, and you click on enter. It shows that we are now inside the local disk. Then we want to go to the folder segment, the users folder segment. So cd means go to, cd users, and then you click enter. It tells us that now we are in users. Then we want to go to the, you can list to check the various options and files that are in this folder, and then it shows us all users, public, desktop, and the likes. Your computer name will be where to go to next. That will be cd. I can type in this fits in, then I press on tab key. The tab key completes it, and you press enter. If you don't want to do that, you can always type it to the end and press enter, but the tab helps to guide you that you're on the right track. In your computer now, you can now go to where the folder is, that is on desktop. You click enter, then you can navigate to the dock folder, that is docking folder in this case, and I'll press enter. In the docking folder now, I want to see if I'm in the right place, so I'll press ls, which means list. It will list the various files that are in there, and you can check with your folder here. I have about 24 items, and if you count here, 1 2 3 4 5 6, by the time you count all through, you have 24 files also here. Now that we are sure that we ensure that the PDBQT files are there, the ligands, they are PDBQT files, and the rec.pdbqt files, because those are the file formats that the terminal understands to run the docking studies. To now run our docking studies here, all we have to put in is the command dot slash vs dot, then you can press tab to complete it, and you press enter. Then it starts running the docking studies. You can expand it, and it has given us the result for our native ligand. That's the first one here, minus 5.3. The speed at which these docking results will be presented and will be executed depends on the capacity of your computer, the RAM, the graphics cards, and the likes. RAM being the most important. So if you have a low memory RAM, you could expect that the docking will take quite some time, but if you have a very good RAM, 8 gig RAM and above, 16 gig RAM, you should complete the docking studies in no time. Then the size of the compounds that you are docking into the active site also matters. The bigger they are, the longer it takes to dock them into the active site. We leave it while it keeps running, and then you can go back soon as it has finished running. You can always go back to Chimera, you close the session, then you open the rec.pdb like we did earlier. Go and view the doc of the result that just came out. Go into tools just like we did earlier in viewing of results, postdoc analysis using either Chimera and Discovery Studio. So go to surface or binding analysis, review the doc, we'll click on the docking folder, and you can see now that Cygwin creates a data 01 folder for you. You click on that data 01 folder and you can see the pdbqt.pdbqt files. This is for the native ligand. If we want to check for our new ligand, first one, that's lig1, it is still running for the other ligand. As soon as it completes it will bring it in here as lig3.pdbqt.pdbqt. But let's check for lig1.pdbqt.pdbqt, and you can see it has given us the docking score for the particular compound that was docked into it. This is a charcoal, and then go ahead and press file. We can save pdb. We save the first pose, and then we can call this rec, then maybe lig1, and we press save. We can go to Discovery Studio. We can close this, and then we can always open the new file that we just saved. That's rec.lig.1pdb, and we press open. It loads it, and then we tell it to step through the entire ligands, and then we can show the 2D diagram. As you can see, it also shows the various interactions that it has with the residues at the active site, and the legend is there. So these are the basic concepts that are actually involved in doc validation and postdoc analysis. And that will be all. Thanks. If you have any issues, please don't forget to reach out to us via WhatsApp. The link is provided in the material and in the details below. Thanks.